Welcome, curious minds. Ever stop to think about, the very roots of fear? That really powerful, almost primal emotion. It can feel overwhelming sometimes, can't it? But it's also crucial for survival. Today, we're going deep. We're journeying into one of the brain's most, well, enigmatic structures, the amygdala. For a long time, our understanding of how it really handles fear, anxiety, it's been a bit fuzzy, let's say. But that might be changing. Our guide for this exploration is some really fascinating research just out from UC Davis Health News. Published October 31st, 2024. This work originally appeared in the American Journal of Psychiatry, a major journal, and it's really pushing the boundaries in neuroscience. So, our mission today to pull out the most important insights from this, frankly, groundbreaking study. This isn't just about satisfying academic curiosity. It's about uncovering new, incredibly precise ways to potentially treat conditions like anxiety and depression. Think of this as your shortcut, giving you the key takeaways on a shift in how we might approach mental health, something that affects millions, right? Okay, so let's unpack this. How could discoveries about this tiny but really mighty part of your brain fundamentally change how we understand and maybe treat these conditions? Now, for many people, the amygdala, it's the fear center. That's the common term. You imagine it lighting up when you're startled or facing something threatening. Strong emotions, basically. And because it's so central, it makes sense. It's linked to things like anxiety, depression. But even though we know it's important, getting a really deep, granular understanding, that's been lacking. So my question is why? For such a key player in our emotional world, why has our scientific grasp been, well, somewhat limited until recently? What was the old thinking, and why wasn't it quite hitting the mark?
Yeah, that's exactly the right place to start. For a long time, the focus in human neuroscience was really on the amygdala's overall size or its structure. Researchers looked for correlations. You know, did variations in physical size, maybe a slightly larger amygdala, or differences in shape link up with someone's chances of having anxiety or depression? The thinking was, maybe these structural differences could predict emotional issues down the line. But as Drew Fox, one of the senior authors on this paper, points out, it's become pretty clear that overall size and structure are, and I'm quoting here, not a good predictor of emotional problems in life. It was a bit like looking at the whole building from the outside when the real issue might be in one specific room, or even the wiring within that room. A macro approach for what seems to be a micro problem. It just wasn't giving us the specific, actionable insights needed for better treatments. The real shift, the paradigm shift, actually started with research in rodents. These studies began to show something fascinating. What we thought of as this single fear center was actually incredibly complex internally. Each subregion within the amygdala contains many different cell types with distinct and sometimes opposing functions. Think of it less like one single unit and more like a diverse neighborhood with different cell types doing very different jobs. Some activating fear, maybe others dampening it. This cellular diversity sparked a powerful new idea. Maybe disorders emerge from alterations in specific cell types with distinct roles, not just a general problem with the whole structure. That realization meant we needed a much, much closer look, a more granular view.
That is a huge shift in thinking. So, if the rodent studies were already pointing towards this incredible complexity, this cellular diversity, well, that presents a massive challenge, doesn't it? Because taking those findings and directly applying them to humans or even other primates is notoriously difficult. So, for ages, the detailed cellular map of the primate amygdala was, as the article says, largely unexplored, a big blind spot in our knowledge, especially concerning human mental health. How do you even start mapping that intricate cellular city inside a primate brain?
Exactly, it's a major hurdle.
And that's precisely the challenge that this team at UC Davis tackled head on. Graduate student Sean Cambridge led this really smart collaboration. It brought together Drew Fox's group from psychology and Cynthia Schuman's lab from the Mind Institute's psychiatry department. Their goal was pretty ambitious: diacellify these distinct cell types within the different subregions of the amygdala, in both humans and non-human primates. And crucially, they wanted to do it based on the unique set of genes each cell type expresses, like finding the specific job description for every resident in that cellular city.
Precisely.
Yeah.
And the technique they used is really at the forefront of neuroscience right now. Single-cell RNA sequencing. Let me just walk you through that quickly. They started with tiny, carefully collected samples from human brains and also from rhesus macaque monkeys. Then, the really key step, they separated individual cells and sequenced their RNA.
Okay, RNA, that's like the cell's current instruction manual, right? What it's
Yeah.
actually doing.
That's a great way to put it. RNA tells us which genes are active being expressed in that specific cell at that moment. It shows the cell's function, its identity. So the why of doing this at the single cell level is huge. It gives you this incredibly detailed snapshot. And the outcome, It lets researchers cluster cells based on their gene expression, identify cell types, and their developmental origin. So you go from seeing just, tissue to seeing distinct populations, neurons sending fear signals, others may be inhibiting them. And this high-res map is exactly what helps identify cell types that are most likely to give rise to psychopathology, mental health conditions. They can specifically look for cell types expressing genes already linked to human anxiety disorders. It's like genetic detective work, cell by cell.
Wow. That's genuinely remarkable. So it's like you said, instead of the blurry forest view, you can zoom right in. You see every single tree, and not just see it, but understand what that specific tree is doing genetically. Its purpose, its activity level, that kind of detail. It must just blow open our understanding of how the amygdala works internally. And this amazing precision, it must have led to some really specific, compelling findings, right? Beyond just mapping cells, what discovery really jumped out as a potential game changer.
Yes, absolutely. One of the most striking discoveries was pinpointing a specific group of cells expressing a gene called FOXP2. Now, FOXP2 itself is interesting. It's known for roles in neural development, even involved in aspects of speech and language development.
Right, I've heard of FOXP2 in that context.
Exactly. So finding it active in a specific cell group within the amygdala, the emotional processing hub, that's already quite intriguing. And what's more, these FOXP2 expressing cells, they have a unique location. They're found right on the edges of the amygdala. These are called intercalated cells.
Intercalated cells on the edges. Why is that location significant? Well, think about it. Being on the edge puts them in a really strategic position, almost like they're sitting at the gateways, controlling traffic between different amygdala regions, or maybe traffic flowing in and out of the amygdala altogether. And this ties directly into their function, which previous rodent research had hinted at. It's been shown, quite convincingly in rodents, that this small group of FOXP2 cells act as gatekeepers. They literally control signal traffic in or out of the amygdala.
Gatekeepers, wow.
Yeah, they can essentially put the brakes on signals, or they can facilitate them. They're a critical control point for the amygdala's overall output.
Okay, hold on. So this isn't just about finding another type of cell. It's finding a cell type perfectly positioned to regulate the whole system's activity, controlling the flow of fear signals, essentially. That really does feel like an aha moment.
It is.
So combining this location, the FOXP2 gene, and this known gatekeeping function from rodent studies, it strongly suggests these intercalated cells could be a really powerful avenue for developing treatments.
It's like finding the master switch, or maybe a set of crucial dials, on the control panel for fear signals. Not just trying to turn down the volume on everything, but finding a specific point of control.
That's a perfect analogy. And the study went even further. They didn't just find these cells in one species. They systematically compared cell types across humans and the macaque monkeys. They were able to identify both similarities and differences between cell types in the human and non-human primate amygdala.
And that comparison is important because
It's crucial for translation. It helped us understand how findings from animal models, which are essential for initial research on things like anxiety or autism spectrum disorders, actually relate to humans. It builds that bridge from the lab bench to potentially helping patients, making sure we're targeting the right mechanisms in people.
Okay, so it's not just fundamental science. It's building a direct road towards, well, actual treatments, precise interventions. What else did they find that points towards those kinds of actionable strategies?
This is where it gets really exciting for potential therapies. The researchers found something else critical about these FOXP2 expressing gatekeeper cells. They also tend to express a receptor that can be targeted by drugs. It's called neuropeptide FF receptor 2, or NPFFR2.
A receptor, like a docking station on the cell surface.
Exactly. It's like a specific molecular antenna or switch. When the right molecule, like a natural neuropeptide or potentially a drug, binds to NPFFR2, it can activate or deactivate the cell, changing its function.
So finding this receptor specifically on these gatekeeper cells, that's huge.
It really is, because this result can guide the development of new treatment strategies. It specifically suggests that drugs designed to activate this NPFFR2 pathway could be a potential treatment target in relation to anxiety-related disorders. This is precision medicine in action, instead of older anxiety meds that might have broad effects across the brain, causing side effects.
Right, the carpet bombing approach versus a targeted strike.
Precisely. Now we have a specific target, NPFFR2, on a specific cell type, the intercalated gatekeepers, known to regulate fear signals. It offers a much more nuanced way to potentially fine-tune that signal flow.
That level of specificity is just, it really contrasts with older approaches, doesn't it? It makes perfect sense what Drew Fox said in the article. Put simply, if we're developing a drug to target the amygdala, we want to know which cell type we are targeting. And this research delivers exactly that. It names the cell type and a specific receptor on it. It's like having GPS coordinates for drug development leading right to the target address.
It truly is a map for future development. And it's important because, let's face it, anxiety is complex. It's a complicated disorder that can present in many different ways. There's no single picture of anxiety. What this kind of detailed research offers is a long-term vision. With this much better, more granular understanding of the cells and pathways involved, it might become possible to identify and target choke points, these critical control nodes that affect large numbers of people who experience extreme and debilitating anxiety.
Choke points. I like that term. Not necessarily a single cure-all, but key places where intervention could provide significant relief.
Exactly. Modulating those choke points could offer a more sophisticated, perhaps more personalized, approach to treatment down the road.
And for you listening, that really is the profound potential here. This isn't just some abstract finding about tiny cells. It feels like a really tangible, hopeful step towards a future where treatments for anxiety, maybe depression too, are much more effective, much more targeted, and crucially, maybe come with fewer side effects. It's about getting right down to the roots of fear at this cellular level. Not to get rid of fear, we need that, but to help us manage it better when it becomes overwhelming, debilitating. To find relief. What an incredible journey we've taken in this deep dive. From the sort of general idea of the amygdala through the power of this single-cell sequencing technique to finding these specific gatekeeper cells and then even identifying a potential drug target, NPFFR2, it really highlights the brain's astonishing complexity. And its elegance, too.
It's absolutely fascinating seeing how these really intricate cellular details are fundamentally shifting how we understand brain function. It just underscores that knowing the details matters immensely, especially when we want to apply that knowledge. It pushes us way beyond just surface-level observations in neuroscience.
Yeah.
This granular view isn't just adding facts, it's genuinely reshaping how we might approach brain disorders.
So here's something to reflect on after this deep dive. Maybe fear isn't just one big monolithic thing. Perhaps, as this research suggests, it's more like a finely tuned orchestra, with specific cells like these gatekeepers playing absolutely critical roles. What could it mean for us, for society, if we could develop ways to precisely help people manage the signals from these key gatekeeper cells, offering real relief from debilitating anxiety, but without just broadly dampening the entire rich spectrum of human emotion, our healthy fear, our joy, everything else? Something to think about. Thank you for joining us on this exploration into the frontiers of neuroscience.